Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about triglyceride synthesis. First, a quick recap of all the metabolism videos we have so far. So in video 5.0, we discuss why it is important for humans to generate or make energy 24/7. And then in video 5.1, we discussed the various ways by which we can make energy, that is ATP, and how we use ATP. In video 5.2, we explain why for humans the most important metabolic highway, the most important metabolic pathway is what we call main street, and that's how we generate ATP from glucose, because glucose is the most important fuel as far as humans are concerned. And that's because the brain and the red blood cells can only use glucose for energy generation, and therefore it is very important for the body, irrespective of what physiological state you are in, early starvation, after a meal, late starvation, whatever, it's always very important that we maintain our blood glucose levels so that ATP can be generated by your brain cells as well as your red blood cells. And then in video 5.3, we talked about the glycolysis part of main street. In video 5.4, to appreciate glycolysis of main street, we talked about some of the defects in glycolysis. What happens if one or more of enzymes of glycolysis is missing, so that you can appreciate the importance of glycolysis. Then in video 5.5, we talked about the PDH part of main street. In video 5.6, we talked about the Krebs cycle part of main street. In 5.7 and 5.8, we talked about the electron transport chain as well as the oxidative phosphorylation part of main street. Then in video 5.9, we talked about defects in the PDH, TCA, ETC, OP part of main street. Then in video 5.10, we talked about chemicals that affect the electron transport chain oxidative phosphorylation, chemicals that we call inhibitors as well as uncouplers. So these videos between 5.2 to 5.10, these videos are all on the most important metabolic pathway, the main street of metabolism in your cells. And once we understand main street, this metabolic pathway, then the rest of metabolism is, you can appreciate much better, you can see the connections much better, because almost all the other pathways are connected to main street. In video 5.11, we talked about the three possible metabolic states that humans can go through, and that is postprandial, after a meal, early starvation, between meals, and late starvation if you have not eaten for more than one day. And in video 5.12, we started with this state, postprandial. So we talked about some of the major pathways that occurs in the postprandial state. And in 5.12, we talked about glycogen synthesis. This is a predominant pathway that happens after a meal in the postprandial state. So this is how we started talking about the postprandial state. In 5.13, another synthesis that happens in the postprandial state is fatty acid synthesis. So in 5.13, we talked about how the body can actually synthesize fatty acids. And today in 5.14, we'll continue the postprandial state and talk about how our body can make triglycerides. Remember, in the postprandial state, when your glucose concentration is high, this is when your insulin levels are high. So your insulin levels are high in the postprandial state. And insulin is a hormone that stimulates synthesis, stimulates a lot of synthesis, stimulates glycogen synthesis, stimulates fatty acid synthesis, and then from there, triglyceride synthesis and protein synthesis. So most of the synthesis pathways that we see happening in the body happen postprandially because insulin stimulates it. The one pathway that it does not stimulate is glucose synthesis, which is called gluconeogenesis. And we'll talk about that in future videos. So as mentioned earlier, it is very important first to understand main street, because the intermediates of main street are used for the synthesis of various molecules, such as your non-essential amino acids and your lipids and so on, and also for the pentose phosphate pathway that we'll talk about in a future video. So since the intermediates of main street are connected to the rest of metabolism, that's why we started talking about main street first. Okay, so let's now discuss triglyceride synthesis, which again happens in the postprandial state. So just to remind you, we are still continuing discussion on the postprandial state. The two most important organs where triglyceride synthesis happens is the liver. So there's a lot of triglyceride synthesis that happens in the liver, as well as the adipose tissue. Okay, so your adipocytes, your fat cells, also make a lot of triglycerides. However, it's very important, and we'll see that later, that although the liver makes a lot of triglycerides, it does not store triglycerides. Okay, so the liver makes triglycerides, and later we'll see these triglycerides as well as other lipids will be sent out as VLDL particles, VLDL. However, the adipocytes, it makes triglycerides and it actually stores the triglycerides as lipid droplets. So you do see a lot of triglycerides, obviously, in the fat cells, but not in the liver. If we do see large amounts of triglycerides in the liver, that's pathological, and that is called fatty liver. Okay, so triglycerides, although they are made in the liver, they are not stored in the liver. The triglycerides are exported as VLDL. However, triglycerides in the adipose tissue are actually stored in the adipose tissue. Understanding lipid metabolism is very important for the healthcare professional because it is associated with cardiovascular disease, obesity, diabetes, as well as fatty liver. So in the previous video, we saw how we humans can take intermediates of main street, such as acetyl-CoA, and then with the help of these enzymes, acetyl-CoA carboxylase and fatty acid synthase, we can then make our fatty acids. And then in the ER, we can elongate these fatty acids that we synthesize. Of all the major biomolecules, carbohydrates, proteins, nucleic acids, and lipids, dealing with lipids has a very unique problem, and that is because lipids are poorly soluble in water, and as you know, our body is mostly water. So for example, a typical fatty acid has a solubility in water of less than 10 nanomolar. But the concentration of fatty acids, total fatty acids, for example, in your bloodstream, in your plasma, is about 100 to 400 micromolar. So remember, this is nanomolar. This is micromolar. Micro is 1,000 times greater than nano. Of course, there is some fatty acids in the plasma that are water soluble, and that concentration is about 8 nanomolar. You can see that is less than 10 nanomolar. So we have some fatty acids that are soluble in our bloodstream, but most of it is not soluble. So how do these fatty acids that are not soluble, how do they exist in your plasma, in your bloodstream? And the answer is the protein albumin. Albumin can bind fatty acids. Albumin is very water soluble, and when the fatty acid binds to the albumin, the whole complex is still very water soluble. So proteins such as albumin, and we have quite a bit of albumin in our bloodstream, can solubilize fatty acids. In fact, the amount of albumin we have can solubilize up to 2 millimolar fatty acids. And you can see the concentration of fatty acids is much less than that. So that's a strategy that the body uses quite often. When we cannot dissolve a certain chemical in water, we use a protein to bind that chemical so that the chemical stays dissolved in water. It's solubilized in water. So this example of albumin solubilizing fatty acid is used very, very often, even for drugs that are hydrophobic. When you take a drug that's hydrophobic, a lot of these drugs, or even hormones such as steroid hormones, they are bound to proteins in your bloodstream, and that's how they travel as soluble chemicals in your bloodstream. So just as albumin helps solubilize the fatty acids in your bloodstream, we also have proteins, for example, fatty acid binding protein, FABP. And these proteins can help solubilize fatty acids in the cytoplasm inside your cells. Now remember, fatty acids, this carboxyl group is ionized at pH 7.4 in your blood or pH 7 in your cells. So that means this carboxyl group is negatively charged, and that negative charge will attract some water. And that's why fatty acids have some water solubility, less than 10 nanomolar. However, we don't have that negative charge in triglycerides. So in triglycerides, you can see the fatty acid is attached to glycerol by an ester. Okay, and esters do not ionize in aqueous solution, so there is no negative charge here. So triglycerides have even worse solubility than fatty acids. So how do we deal with triglycerides? Well, in the bloodstream, we'll see that these triglycerides are actually part of particles. Okay, so these are not water-soluble substances. These are particles we call lipoproteins. Okay, so we are talking about particles called lipoproteins. And that's how triglycerides and many other lipids travel in the bloodstream. Inside the cell, we will see that these lipids are made in the endoplasmic reticulum membrane, and they are either stored as lipid droplets or secreted as VLDL. So let's look at this part in a little bit more detail. So the enzymes that synthesize triglycerides, you can see they are bound to the membrane of the endoplasmic reticulum, the smooth endoplasmic reticulum. So this is the membrane of the smooth endoplasmic reticulum. This is the cavity of the SCR. And you can see the lipids that are made, the triglycerides that are made, they are located in the hydrophobic part, you see, inside the membrane. As more and more of these triglycerides and other hydrophobic lipids like cholesterol esters are synthesized, they accumulate and they form what is called a lipid lens in the smooth endoplasmic reticulum membrane. So you can see this newly synthesized triglycerides as well as cholesterol esters accumulate and form this sort of a droplet. So with the help of proteins such as perilipin and cypin and several other proteins. What happens is you can see that the droplets, these lipid droplets, will bud off from the SCR membrane. And then in adipocytes, these lipid droplets will be stored in the adipocytes, in the cytoplasm, as lipid droplets. And you can see the lipid droplets has an outer phospholipid membrane, monolayer membrane, as well as some proteins such as perilipin. And the inside of the lipid droplets will consist of all the hydrophobic lipids, such as your triglycerides and your cholesterol esters. So this is what we see in adipocytes. In hepatocytes, in the liver, we will see that the liver will make a protein. In this case, for VLDL, it will make the protein ApoB100. And this protein is made in the rough endoplasmic reticulum. This protein will then combine with the lipid droplet. Okay, so these two will combine in the liver cells. And the protein and the lipid droplet will form this structure here that will then bud off from the smooth endoplasmic reticulum and form what we call a lipoprotein particle, an immatured lipoprotein particle. This newly made lipoprotein particle is then transported to the Golgi, and in the Golgi, the lipoprotein particle, in this case the VLDL, is matured. Okay, it takes in more apoproteins, maybe more lipids, and then from the Golgi, it will be excreted out of the liver cell. Remember, it's not stored in the liver cell. So VLDL is excreted out of the liver cell by exocytosis, and the VLDL then enters the bloodstream. Okay, so unlike adipocytes that store their lipids as lipid droplets, the liver makes the lipids, then exports it out into the bloodstream. So now let's look at the key steps in the synthesis of triglycerides. Remember, triglycerides are also called triacylglycerols, that we abbreviate TAG. So let's look at how these triglycerides are made in your smooth endoplasmic reticulum. Just to remind you, triglycerides have two parts, your glycerol and your fatty acids. So here is the ER membrane, the outer monolayer, and this is your inner monolayer of your ER membrane. So this would be this here, and this would be this. Of course, the head will be on the other side. Okay, so this is your smooth endoplasmic reticulum. And this space here would be your cytoplasm. So what we need is this building block. This is glycerol 3-phosphate. And glycerol 3-phosphate can come from the reduction of dihydroxyacetone phosphate. You can see this CO group is reduced to an alcohol. And dihydroxyacetone phosphate, if you remember, is an intermediate of glycolysis. Now, we can also get glycerol 3-phosphate from glycerol by phosphorylating this OH group here. In the next step, we add a fatty acid in the form of fatty acyl-CoA right here. And now we get, so glycerol 3-phosphate, which is water-soluble, now becomes lysophosphatidate, which is not water-soluble, so that's embedded in the smooth endoplasmic reticular membrane. So we see the fatty acid connected to the glycerol phosphate to form lysophosphatidate. And this reaction is catalyzed by an enzyme, glycerol 3-phosphate acyl transferase, that is found in the mitochondria as well as in the ER. In the next step, another enzyme, acyl transferase, that's embedded in the ER membrane, adds another fatty acid, and this time to the middle carbon here. So now we have another fatty acid attached, and this gives us this product here called phosphatidic acid or phosphatidate, also called diacyl glycerol phosphate. Okay, diacyl, 2 fatty acids, glycerol phosphate. In the next step, the phosphate group is removed from phosphatidic acid or DAG phosphate, and that's done by phosphatase, an enzyme phosphatase. So that removes the phosphate group, and now we get diacyl glycerol. And you can see the phosphate group is gone, and we have our 2 fatty acids here connected to glycerol. So this is called diacyl glycerol. And in the last step, another enzyme, transferase enzyme, will transfer a 3rd fatty acid now to this hydroxyl group at this carbon, and that gives you another fatty acid. So 1 fatty acid, 2 fatty acids, 3 fatty acids attached to the glycerol part, and that gives us a triglyceride or triacyl glycerol. And as we mentioned earlier, this triacyl glycerol, in case of the liver cell, will be exported as VLDL. In case of adipocytes, it will be stored as lipid droplets. So that is how your liver cells as well as your adipocytes synthesize triglycerides. In the next video, we'll talk about how we synthesize the amphiphilic lipids, the surfactants. So until the next video, thank you for watching this video. Have a wonderful day, and hopefully we'll see you soon.